Have you ever pondered the intricacies of DNA replication? This is not just a biological phenomenon, but a marvel of nature that ensures the continuity of life. DNA replication is the process where an organism duplicates its DNA, creating two identical DNA molecules from a single original DNA molecule. This is a vital process for life, ensuring that when cells divide, each new cell has an exact copy of the DNA present in the original cell. In this fascinating dance of life, enzymes and proteins are the choreographers. They facilitate and control the replication process, ensuring that it runs smoothly and accurately. Without these tiny powerhouses, DNA replication would not be possible. They act as the catalysts, speeding up the reactions, breaking and forming bonds, and ensuring the correct sequence of bases. Now that we understand the basics, let's delve deeper into the role of enzymes in DNA replication. The first group of enzymes we'll discuss are the nucleases. These are the molecular scissors of the DNA world, cleaving phosphodiester bonds between nucleotides. They come in two main types: exonucleases and endonucleases. Exonucleases nibble away at the ends of the DNA chain, moving either from the 5 to 3 direction or the other way around. Endonucleases, on the other hand, prefer to cut in the middle of a DNA sequence. Some are selective, choosing to cleave either DNA or RNA, thus earning the nicknames DNAs and RNAs respectively. But nucleases aren't just about breaking things apart. They've become indispensable tools in the modern field of gene editing. The CRISPR genome editing technique, for instance, employs a special nucleus called Cas9 to precisely cut and alter DNA sequences. So, nucleases play a crucial role in DNA replication, but they're not the only ones. Another key player in DNA replication is the restriction endonuclease. These are highly specialized enzymes that cleave DNA into fragments at or near specific recognition sites within the molecule, known as restriction sites. To break down the DNA, restriction endonucleases make two incisions, once through each sugar phosphate backbone of the DNA double helix. Imagine a pair of molecular scissors, cutting precisely at designated points. That's essentially how restriction endonucleases work. They recognize a specific sequence of nucleotides, usually 4 to 8 bases in length, present in the recognition site. Once they identify this sequence, they produce a double-stranded cut in the DNA. This action is not random, but rather a highly targeted process, ensuring that the DNA is cut at the correct location. This precision is crucial in DNA replication, where accuracy is paramount. Restriction endonucleases, with their precise cutting ability, contribute significantly to DNA replication. Apart from nucleases and restriction endonucleases, several other enzymes and proteins aid in DNA replication. One of them is DNA polymerase. It's the key player that adds new nucleotides to the growing DNA strand, ensuring each new cell has a perfect copy of the genetic information. But before DNA polymerase can get to work, we need helicase. Helicase unzips the double-stranded DNA, breaking the hydrogen bonds between the base pairs and creating two single strands ready for replication. But it's not a two-man show. We have enzymes like primase, which creates a primer to kickstart the replication process. We also have ligase, which seals up the fragments of DNA in the lagging strand to create one continuous strand. And let's not forget the protective proteins, like single-strand DNA binding proteins, which prevent the single strands from rejoining and protect them from degradation. These enzymes and proteins work in harmony to ensure accurate and efficient DNA replication. DNA replication is a complex yet fascinating process made possible by a team of enzymes and proteins. We've delved into the intricate roles of nucleases, enzymes that cleave phosphodiester bonds between nucleotides. There are two types: exonucleases and endonucleases, work from different ends, with the former cleaving from the extremities and the latter acting on the central region of the targeted nucleotide. We explored the particular role of restriction endonucleases, which cleave DNA into fragments at or near specific recognition sites, making 2 incisions through each sugar phosphate backbone of the DNA double helix. This selective cleavage is vital for gene editing and other scientific advancements. We've also touched on the roles of other pivotal enzymes and proteins that work in concert to ensure precise and accurate DNA replication. This intricate dance of molecular interactions is what makes life possible. Next time you marvel at the complexity of life, remember the enzymes and proteins diligently working to replicate DNA, one nucleotide at a time.